My name is Erkan Karakas. I'm an assistant professor at the Department of Molecular Physiology and Biophysics. I'm also a member of Center for Structural Biology. The focus of my research is understanding the molecular mechanism of communication between endoplasmic reticulum and mitochondria. The reason I'm interested in this ER-mitochondria communication or communication between organelles in general is that this is a very new field. We only started to understand how they are mediated and they function within the last decade. And there are many questions that we don't know the answer yet. So what I want to do is basically contribute to understanding of this fundamental cell biological process. The major experimental systems we use are X-ray crystallography and cryo-electron microscopy. We had a very major recent advance in this field, and now we can reach very high resolution structures using the cryo-electron microscopy. It actually started after the biochemistry classes I took in my college year when we were studying protein chemistry, and I was immediately attracted to it. I was fascinated by the fact that individual amino acids can form a chain, and this chain folds around itself to form a protein that functions, that does diverse things. They can catalyze reactions, chemical reactions. They can carry molecules from one place to another inside the cell. They form gates across the membranes and selectively permeate certain chemicals. So it was just fascinating, and to study that using structural biology felt like solving puzzles. You basically solve puzzles using these different methods, and at the end you obtain the structure of this protein, and that's the great feeling or great satisfaction to know that when you solve the structure for the first time, you're the first person in the world to see how that protein is shaped. As a new faculty, we need more private funding because getting federal funding at the beginning is more difficult compared to more established scientists, because they already have their track record as an independent scientist, and they also have preliminary data that show that they can produce data, and the federal fundings are usually fund more guaranteed research. But as a young investigator, we don't have yet. We haven't established ourselves as an independent investigator. We don't have usually, at least at the beginning, we don't have enough preliminary data to show that we can do this. So therefore, the private funding is essential so that we can produce the preliminary data, we can publish our work, so that we can, when we apply for federal funding, we actually have already a track record, enough preliminary data, so that it would increase our chance of getting federal funding. The colleagues were very important for me. There is a really nice structural biology community here, and also other colleagues in different fields. It's very diverse, and it's often very easy to find someone to collaborate on. Even when we have something in our mind, we can just find someone next door to talk to and even collaborate whatever we need, even though it's out of our field or immediate field, or if we need a certain instrumentation, we can easily find that at Vanderbilt University. It usually takes time to get some exciting or breakthrough moments, but to get there you have to persistently work and just be patient with the outcome of the research. And also at the same time, I find it important for young people to celebrate small accomplishments. like to properly run an experiment with the appropriate controls. You're not gonna have a major breakthrough every day, but if you wait for that, it may be it may be depressing sometimes. So it's important to to be able to celebrate those small accomplishments too, so that you have energy or morale to push forward for the next big thing. I had my undergraduate study in Middle East Technical University in Turkey. I studied molecular biology and genetics there. And after that, I came to United States to have my PhD in Stony Brook University. There I worked with Caroline Kisker and trained as an X-ray crystallographer, and I was working on structural characterization of nucleotide repair enzymes. And after completing my PhD, I went to Cold Spring Harbor Laboratory to work with Hiro Furukawa, and I made a major change in my field. This time I started working on neuroreceptors, ion channels, specifically NMDA receptors, which are important in synaptic transmission in mammalian brain, and it's essential for learning and memory formation. And I was able to solve the first crystal structure of intact NMDA receptor, and that was a major breakthrough in my research during my postdoctoral work. And then I came to Vanderbilt University as an assistant professor, and I've been here since.